Okay, so we can start. What we're going to do today is we're going to start a new section, and we'll begin this section with a discussion of the respiratory system. And we're going to talk about the structure and function of the respiratory tract today. And we're going to begin with the concepts of its function. And some of these will be familiar to you, some of them may be a little more arcane, but gas exchange is fundamentally what we're doing with the lung and the respiratory system. And what we mean by gas exchange is oxygen is diffusing out of the air and into the blood. And so we're going to see that we have these specialized structures called alveoli where we get this diffusion. So the first kind of gas exchange we're going to talk about is diffusion of oxygen from the air into the pulmonary capillaries. But the other form of gas exchange that we see here is carbon dioxide, which is moving from the blood in the pulmonary capillaries to the air sacs. Now, oxygen is in the air around us. About 21% of the air around us is oxygen. The remainder is pretty much nitrogen. So we're breathing that in, and we're going to have a high concentration of oxygen in the air sac and a lower concentration of oxygen in the blood, because this blood is coming back from the systemic circuit. It's come back through the right side of the heart. It's been pumped to the pulmonary capillaries through the right ventricle, or by the right ventricle, and this blood now is going to be oxygen poor. And we can now use the gradient of oxygen in the air versus the oxygen in the pulmonary capillary to allow oxygen to diffuse into the pulmonary capillary. There's nothing magical about this. This is just a diffusion gradient. And oxygen is going to diffuse from a region of higher concentration to lower concentration. The other side of that coin is that the other form of gas exchange is going to be carbon dioxide. And carbon dioxide is going to diffuse out of the blood into the air sac. But of course, where does the carbon dioxide come from? The carbon dioxide is coming from as a waste product. It's a waste product produced from the final electron acceptor of the cytochrome C, and we generate carbon dioxide. That carbon dioxide now is then soluble in water, and it can form carbonic acid, which then dissociates into bicarbonate and hydrogen ion. It's this bicarbonate that's functioning as a buffer. It's maintaining the blood pH between 7.35 and 7.45. If you take some chemistry, you'll recognize that bicarbonate is a terrible, terrible buffer at those pH ranges. It's much, much better down around 4.5 or 5.5. But we have so much carbon dioxide in the system that we can still use bicarbonate as a fairly effective buffer, even though it's not in its ideal range. And we use bicarbonate to maintain blood pH between 7.35 and 7.45. We'll also see that this reaction of carbon dioxide plus water yielding bicarbonate and hydrogen ion can run in the other direction, but either way, it's slow. If we're simply using the law of mass action in chemistry, it's really slow. So what we're going to see later on in this unit is that there's an enzyme called carbonic anhydrase. And carbonic anhydrase will increase the rate of this reaction by about 1,000,000-fold. And we'll see that out in the tissues, it's generating bicarbonate. And then in the lung, it's generating carbon dioxide. This carbon dioxide then can diffuse from the blood vascular compartment, the pulmonary capillaries, into the air sac, and then go to the outside world. So those are the functions, and we'll get down into some of the anatomy and how it all fits together with the functions. But first, we have to talk about the overall anatomy of the respiratory tract. And there are four major characters we're going to talk about in this region, which I'm going to label the conducting zone. When you see this term conducting zone, think of it as a series of tubes. They're just nothing more than tubes that are delivering air down to the business end of the system, which is going to be the respiratory epithelium. Now, when we talk about these structures, we're going to talk about the larynx. This is the voice box. We'll mention it just briefly. The trachea is a single tube leading into two bronchi. The singular of bronchi is bronchus. The bronchi then further branch and branch and branch into bronchioles. And we'll see that we start out with one tube as the trachea. And before long, these bronchi and bronchioles have branched thousands of times to increase the total surface area of the terminal regions of this structure, which are in the respiratory zone. The respiratory zone is composed of an alveolar epithelium. When you see this term alveolus or alveolar, think air sac. And the alveolar epithelium is going to provide the epithelium across which gases can diffuse. These gases will diffuse across the alveolar epithelium and diffuse across the pulmonary endothelium to get into the blood. Oxygen diffuses in, and carbon dioxide, of course, diffuses out. Then we're going to spend some time talking about the bronchioles of the conducting zone and how these bronchioles are under autonomic regulation. The autonomic regulation of the bronchioles resides in epinephrine and acetylcholine. This should look familiar to you. These are more or less the molecules we talked about with autonomic regulation of the heart. But here the story is going to be slightly different. We're going to see here that epinephrine is going to function through a beta-2 receptor. Remember, the heart was beta-1. This is a different kind of receptor. And instead of what we saw in the heart working through G-alpha-S, this is going to work through G-alpha-I. And the G-alpha-I now is going to inhibit adenylyl cyclase. And by inhibiting adenylyl cyclase, we're now going to cause muscle relaxation. So smooth muscle relaxation results from the activity of epinephrine. Now, there's a subtle difference here. When we talk about norepinephrine, that's a proper neurotransmitter. That proper neurotransmitter is being released by the postganglionic sympathetic neuron. We don't have that kind of innervation in the lung. Instead, what we do is we rely on epinephrine, which is being released from the adrenal medulla. So in this case, rather than talking about a neurotransmitter, we're actually talking about a hormone being released from the adrenal medulla. But we do have proper parasympathetic innervation of the smooth muscle in the bronchioles. That proper parasympathetic innervation will have postganglionic parasympathetic neurons releasing acetylcholine. That acetylcholine will bind to an M3 receptor. Now, this M3 receptor does something different than we talked about for M2s in the heart. Here, M3 works through something called G-alpha-Q. It leads to calcium release from internal stores, and this calcium release from internal stores leads to smooth muscle contraction. So if you think about this for a minute, what you see is when you're under sympathetic drive, when your sympathetic system has its dominant effects, then the bronchioles are dilated because the smooth muscle is relaxed. Because the bronchioles are dilated, you know from Poiseuille's law that the flow is going to be greater. The Poiseuille's law tells you that flow is proportional to the radius to the fourth power. So we don't have to change the radius very much here, and we increase the flow by a whole lot. So epinephrine is going to increase the flow by relaxing or increasing the diameter or radii of those bronchioles. The other side of the coin is that acetylcholine, through M3 receptors in the parasympathetic branch of the autonomic nervous system, this is going to cause smooth muscle contraction. It's going to reduce the volume of the conducting zone. Now, at first blush, you say, why is this set up this way? You can sort of see the one for epinephrine. You want to relax your undersympathetic drive. You want to relax those muscles, increase the diameter so you get better flow of oxygen and air down into the alveoli. But why is it when you're sitting at home relaxed and watching TV that you activate the parasympathetic branch? I don't have a really good answer for it, but I think what's happening is when you activate the parasympathetic branch and you constrict those muscles, now what you're doing is you're reducing the overall volume of the respiratory tree that is dedicated to simply the respiratory zone. Remember, the respiratory zone are just tubes. They're just pipes. And so when you're under parasympathetic drive, then you reduce the volume that's dedicated to just the tubes. And a greater overall proportion now of the whole tree can now be dedicated to the business end, which is going to be the respiratory epithelium. But let's start here with just a list of things that we talk about functions of respiratory system tract. And the first one is going to be oxygen exchange. touched on this, but I think it's worth looking at it again. I'm going to draw this structure up here as though it were a box, but in fact, this structure we call the alveolus as part of the respiratory zone. Normally, these epithelial cells would be organized as a kind of sphere. It would look like a grape on a bunch of grapes, but I'm going to draw it as a box because it can tell us the same story. This is the air. If you stood on the epithelial cells here in the alveolar sac, you could walk from one epithelial cell to the next, find your way out of the respiratory epithelium into the conducting zone, and then go all the way up to the outside world. But this region contains these cells called epithelial cells, which in this context get another name called pneumocytes. Those pneumocytes are exposed on their apical surfaces to the air. But of course, as an epithelium, there's a basal lamina. And that basal lamina sits right next door to the basilar surface of endothelial cells. Remember, the pulmonary capillary is carrying deoxygenated blood, but it's carrying deoxygenated blood on erythrocytes. If the flow is in this direction, then what we're seeing is that deoxygenated, let me do it this way, O2 poor blood comes in, as it's flowing by the alveolar space. Coming out the other side, we have O2 rich blood. And the dwell time, the period of time when these erythrocytes are closely opposed to the epithelial cells, this time is less than 0.75 seconds. That's when we're sitting quietly. If you're exercising really hard, this can get down to 0.25, 0.3 seconds. All of that is to say that within this very brief period of time, we have to load up the hemoglobin on the erythrocytes with oxygen. And we can see that the oxygen now, which is in air, is in higher concentration than the oxygen in the blood returning from the tissues. That oxygen has been consumed in the tissues and converted into carbon dioxide. So this is oxygen poor. So now we have a gradient. And the oxygen can diffuse across the epithelial cell, diffuse across the extracellular space, diffuse across the endothelial cells, and enter the blood. We're just using the gradient. The oxygen is moving from a region of higher concentration to a region of lower concentration. And it's in higher concentration in the air than it is in the blood. It comes to equilibrium before it leaves, so we end up with oxygen-rich blood. Now, when we look carefully at this, and we'll talk about this in more detail, there's a very, very thin layer of water over the apical surfaces of those epithelial cells. And the oxygen first has to dissolve in that very thin layer of fluid. Then it has to pass through or between the epithelial cells, pass into the extracellular space, pass through or between the endothelial cells to get into the pulmonary capillaries. Now, one of the things I want to point out here, because it's something that's affecting our lives right now, is the idea of pneumonia. Pneumonia is the buildup of fluid in the alveolar space or between alveolus and capillary. This is sometimes known as interstitial pneumonia. What's happening here is that when we begin to get inflammation in the lung, we begin to get inflammation in the alveolar sac, and we begin to build up a layer of fluid air, we're increasing the diffusion path. The length of the diffusion path is getting longer. The longer the diffusion path, the longer time it takes for the oxygen to come from the air in the alveolar sac and get into the blood vascular compartment. If this diffusion distance gets to be too great, we simply cannot adequately oxygenate the erythrocytes. So this is where pneumonia comes into play. Pneumonia occurs when we get inflammation in here. Sometimes it's from bacterial infection, sometimes it's from viral infection, sometimes it's a response of our immune system to those various bacteria and viruses. And that's what we think is going on in many cases with COVID. We get this massive immune response called a cytokine storm, and it builds up fluid layers here in the alveolar space and in the space between the pulmonary capillary and the epithelial cells. I should point out that these distances are exquisitely small. The distances taken from the apical side of the epithelial cell across the extracellular space, across the endothelial cell, these distances are about 0.1 to 0.2 micrometers. Very thin, because as we talked about with the erythrocytes, what we're trying to do is limit diffusion distances. And these exquisitely thin cells with an exquisitely thin layer of water over them now limit the diffusion distances between the air in the alveolar sac and the erythrocytes in the pulmonary capillary. But this is the business end of the system. This is where gas exchange is occurring. The gas exchange we've of is oxygen diffused in the pulmonary capillary. But as the blood's coming back from the tissues, it's going to come back as CO2 rich blood. So the CO2 rich blood now is passing by the alveolar capillaries. There's now another gradient where carbon dioxide's concentrations are greater in here than they are in the alveolar sac. So CO2 diffuses from the pulmonary capillary across the endothelium, across the extracellular space, across the endothelium, and out into the alveolar sac, and then you breathe it out to the outside world. Typically what will happen is the concentration of carbon dioxide as it's passing through these pulmonary capillaries will now fall as from the time it comes in to the time that it leaves, because it's diffusing down its concentration gradient. And that's the fundamental idea that we're going to talk about in more detail about how oxygen diffusion and gas exchange occurs in the lung. So any questions to there? That's the BIS.
So we talked about how oxygen binds to the iron atoms on the erythrocytes. What is CO2 doing?
Oh, great question. Okay. Let me do it up here. We're going to talk about this in more detail, but we may as well do it now. Something like 70% of CO2 in blood is carried as bicarbonate, HCO3 negative. I think it's something like 5 or 10% is bound to the hemoglobin, and the remainder is basically just in solution. But most of it, the lion's share of it, is coming as bicarbonate. And so what we see is in the tissues, carbon dioxide plus water gives rise to HCO3 negative plus a hydrogen ion. We'll see later that the hydrogen ion is going to bind to the hemoglobin, and most of the CO2 is going to be carried as bicarbonate. But this reaction is reversible. it's especially reversible when we talk about enzymes, and we'll get into that. But as we can take CO2 and water and make bicarbonate, this is what happens in the tissues. In the lung, we take bicarbonate, HCO3 negative, and we convert this back to CO2 plus water. So your question is spot on. We're carrying oxygen on hemoglobin, but we're carrying carbon dioxide predominantly as bicarbonate dissolved in the blood. And because we have these clever enzymes, we can rapidly make bicarbonate and hydrogen ion in the tissues. And then back in the lung, we can take the bicarbonate, convert it back into CO2, and let CO2 then diffuse back out of the blood into the out into the alveolar space. Okay? Anything else? Okay, let me get rid of this then. And now let's talk about the anatomy of the respiratory system. So we'll just take this portion of the board and label it as anatomy of the respiratory tract. We're going to start out here with a structure sort of funnel shaped called the larynx. We know this commonly as the voice box. It's the voice box because within the larynx there are these flaps of tissue known as vocal folds. When I'm making sound, when I'm making language, when I'm speaking, I'm vibrating those vocal folds. We have exquisite control over that vibration. There's some individuals who are well trained who can vibrate these vocal folds at a precise frequency to hit a particular note when they're singing. So if you ask a well-trained vocalist to hit a particular note, hit a 440A, they can vibrate that vocal fold at precisely that frequency to make that note. I can't hope to do that. I'm lucky to speak. But these vocal folds are essential for us making sound. And the way we control them is we control the movement of air out of the lung, out to the outside world, and that's how we're controlling the movement of those vocal folds. The larynx leads into a structure known as the trachea. The trachea is a single tube. One of the characteristics of the trachea is that it's reinforced by these cartilage rings. So hyaline cartilage forms these reinforcing rings, and the reason we need that reinforcement is, we'll see a little later, that the way we breathe in is we generate a slight vacuum inside our lung. By generating a slight vacuum inside our lung, there's a greater pressure in the outside world than there is in the inside of our lung, so that pressure differential generates a flow. But if we didn't have these supporting rings, when we go to breathe in, the pressure now would tend to collapse the trachea. So we reinforce the soft tissues of the trachea with these rings of cartilage. And then the single tube of the trachea branches into structures called bronchi. Bronchi is plural of bronchus. So we have these two bronchi branching off from the single tube of the trachea. These two are going to be lined with, or be ringed with, I should say, hyaline cartilage. But these bronchi then branch and branch and branch. There's a tremendous amount of branching here. Each of these tubes branches over and over and over again. One set leads to the left lung, and the other then will lead off to the right lung. The main idea here is that these bronchi branch into bronchioles. Bronchiole is IOLE, is a suffix in Latin which means small. So these are little bronchi. So we're branching and branching and branching until these branch thousands, literally thousands and thousands of times, and we end up at the very end of the bronchi, the structure I'm going to label here as the alveolus, or the air sac. So this entire system, the larynx, the trachea, the bronchi, and the bronchioles. Taken together, these are said to be the conducting zone. These are a series of tubes delivering oxygen to the alveoli and delivering CO2 from alveoli to the outside world. So what we're doing with this conducting zone is basically providing a highly branched series of tubes to bring the air down to the alveolus. That air then delivers oxygen into the pulmonary capillaries, and from the pulmonary capillaries we get carbon dioxide going out the other direction. Let's look at a couple of pretty pictures of this. The first is just an overall image of what I drew on the board there. But it emphasizes, I think, much of what we want to think about, which is to say, we start out here with the larynx. That's a single structure that then opens up into the trachea. The trachea then branches into the bronchi, and these bronchi then further branch into the bronchioles. And down at the very ends down here will be the alveoli. This is a more accurate depiction. These alveoli are like grapes in a cluster of grapes. The diameter of each one of these alveoli is probably no more than 80 micrometers or so. But they're multicellular structures. They are composed of a single layer of epithelial cells. And those single layer of epithelial cells are lying adjacent to the endothelial cells of the pulmonary capillaries. Now, this is a figure with a bunch of numbers on it. Don't worry about the numbers. But I like the figure because it's showing how we branch from a single tube, the trachea, and within a short period of time, we end up with literally millions of these alveoli. And it shows here that we start out with the trachea, then we branch into the true bronchi, and then we have the bronchioles, the major bronchioles, and these branch and branch. By the time we get to the terminal bronchioles, we have 60,000 of these things. And then these further branch until finally we get down to the air sacs, and we have like 8 million of them. Some places you'll read about this, and they'll talk about the overall area of the alveolar sacs being roughly the size of half a tennis court. So through this branching system, we're increasing the surface area of the alveolar sacs, excuse me, where the oxygen exchange actually occurs. Now, in this particular figure, they describe the respiratory epithelium as being the respiratory bronchioles, the alveolar ducts, and the alveolar sacs. But we're going to think about the alveolar sacs as being predominantly the site where gas exchange occurs. This next picture is a picture which is going to lay out the next description. This is a figure showing the terminal bronchioles. And one of the things I want you to see from this figure is how these bronchioles contain around them, they have these straps of smooth muscle. We're going to talk about how the straps of smooth muscle are going to regulate the diameter or radii of these bronchioles. But this is showing how the bronchioles end in this cluster of grape-like structures called the alveoli. Each one of these spherical structures is meant to be an alveolus. In reality, this will probably be no more than about 80 micrometers in diameter. And what we're looking at here is we're looking at the basilar surface of those epithelial cells. And that's right adjacent to the basilar surface of the capillaries of the pulmonary capillaries. Now, this color difference is trying to display the idea that deoxygenated blood is coming in through these pulmonary capillaries. This is from the right side of the heart. So the right side of the heart is pumping deoxygenated blood to the lung. And then these capillaries, as they course over the alveolus, will then receive oxygen, and this is demonstrated here by turning from blue to red, demonstrating how we're oxygenating the blood in the pulmonary capillaries as it crosses the alveolus, and then is gathered by blood vessels to take this back through the pulmonary veins back to the left side of the heart. But this is showing the site of oxygenation. Where we're going to go now, though, is we're going to go upstream a bit. We're going to start talking about this upstream region where we have these smooth muscle straps. And we're going to begin to think about how we can regulate the diameter of those tubes by using those smooth muscle straps. So we can imagine all of these bronchioles are surrounded by smooth muscle. So this is meant to be smooth muscle. And we can say here that the smooth muscle regulates the diameter / radius of the bronchioles. So the relative diameter of this conducting zone, the relative diameter of the tubes in the conducting zone now can be modulated by the smooth muscle. And of course, whenever you hear the term smooth muscle, you should immediately be thinking autonomic nervous system. So let's spend a little bit of time thinking about autonomic effects. On the bronchioles. So we'll take this portion and we'll label this as autonomic effects on bronchiolar smooth muscle. And I'll tell you at the outset, we're going to be talking about a whole host of these G protein coupled receptors. So let's imagine in orange we have that strap-like muscle. Let's take an example of one of these muscle cells. So if this is a smooth muscle, we don't have time to get into all the details here, but it obeys many of the same rules that we talked about with skeletal muscle. There's some subtle differences, but there are in fact Z lines or Z discs. And attached to the Z discs will be actin-thin filaments. And between those actin-thin filaments, again, will be a myosin-thick filament. If we'd get down into the weeds of this, we'd find that the myosin-thick filaments have myosin whose characteristics are different from what we saw in skeletal muscle, and the thin filaments have differences as well. But the long and the short of it is still the same, that the myosin-thick filaments will have myosin heads that ratchet along actin-thin filaments. So they're going to be ratcheting along these thin filaments. They're going to cause the muscle to contract. So smooth muscle contraction shortens the length of those smooth muscle cells. And you can imagine what's going to happen up here as these are wrapped around the bronchioles. When you shorten those smooth muscles, when you contract the smooth muscles, you're going to on those bronchioles. And by clamping down on the bronchioles, you're going to change the radius. You're going to reduce the radius. And you'll remember from Poiseuille's law that the flow is proportional to the radius raised to the fourth power. So we don't have to change that diameter radius very much, and we're going to affect the flow. So now let's think about the control systems for this. Go up to the upper right-hand panel first. We're going to see that that smooth muscle is in fact under autonomic control. And the autonomic control here is going to be slightly different, but familiar to what we talked about with the heart. So let's first talk about the sympathetic response. Let's first talk about the sympathetic response of bronchiolar smooth muscle to epinephrine. Here's one of the first differences. In the heart, we talked about postganglionic sympathetic neurons releasing the neurotransmitter norepinephrine. There's only a methyl group difference between norepinephrine and epinephrine, but it turns out epinephrine is released from the adrenal medulla. So there isn't much in the way of what we would consider formal sympathetic innervation of the lung. The sympathetic effect, the autonomic sympathetic effect, is arising from epinephrine released almost as a hormone. Then, if we look at one of these smooth muscles, so if this is one of these smooth muscle cells, there's a transmembrane protein here. It's a G protein coupled receptor, so it's going to cross the membrane 1, 2, 3, 4, 5, 6, 7 times. This will be the epinephrine receptor. When epinephrine binds to the epinephrine receptor, we now bring into play those old friends of ours, the heterotrimeric GTP binding protein. So it's an alpha, a beta, and a gamma. I'll remind you that the alpha and beta and gamma all are cuddled up together as long as the GDP is attached to the alpha subunit. But when the epinephrine binds the G protein coupled receptor, it opens up a site in the cytoplasmic solid. Just as before, this alpha subunit nestles into that binding site that's been opened by the binding of the neurotransmitter. That leads to the exchange of GDP for GTP. The alpha subunit comes loose from its mates the betas and gammas. And it goes off and activates a transmembrane protein here called adenylyl cyclase. Now, I should emphasize here that this particular alpha subunit is G alpha I. So it's not going to activate the adenylyl cyclase. This G alpha I is going to inhibit adenylyl cyclase. This leads to a drop in cyclic AMP levels. The drop in cyclic AMP levels leads to smooth muscle relaxation. So the effect of norepinephrine on bronchial or smooth muscle causes smooth muscle relaxation, and that's in contrast to what we saw with the autonomic effect in the heart. And the autonomic effect of the heart, remember, the beta-1 receptors caused the beta-1 receptors called cardiac muscle contraction and increased heart rate. So it turns out that these epinephrine receptors are a slightly different variant of those adrenergic receptors. And these epinephrine receptors get a new name. They're called beta-2 receptors. The beta-2 receptors predominantly, not uniquely, but mostly they're found on this bronchiolar smooth muscle, and we can see how this is going to be used pharmacologically here later. But these beta-2 receptors, the binding of epinephrine to the beta-2 receptors leads to smooth muscle relaxation. Hold on to that idea. Then down here on the other panel, over here, we're going to tell the same story. Let me erase this and we'll tell the same story with another smooth muscle cell. It could be the same smooth muscle cell. But in this case, we're going to talk about the parasympathetic effect on bronchiolar smooth muscle. So this will be the parasympathetic effect on bronchiolar smooth muscle. So now, this box, just as we showed in the upper right, this is now an example of bronchiolar smooth muscle. Now, again, there's a G protein coupled receptor. Crosses the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. This is said to be an M3 receptor, which of course is a type of muscarinic receptor. And you remember that the muscarinic receptors bind acetylcholine. So in the lung, in the bronchiolar smooth muscle, there is proper parasympathetic innervation. There will be a pulmonary ganglion where we'll have the postganglionic parasympathetic neurons. Those postganglionic parasympathetic neurons will now innervate the bronchiolar smooth muscle. They'll release acetylcholine. Again, they work through, this of course is a type of GPCR. So of course, it's going to work through a heterotrimeric GTP binding protein. Alpha, beta, and gamma. It too contains a GDP. And when the alpha subunit nestles into the binding site, which has been opened up by the binding of acetylcholine to this M3 receptor, then the GDP comes off and is exchanged for GTP. Now we have this alpha subunit with GTP attached. And this alpha subunit goes out and activates a protein called phospholipase C. I'm not going to go through all of the steps, all the pathways, but suffice it to say that phospholipase C gives rise to the release of calcium from internal stores. This calcium will be released from the endoplasmic reticulum, which in this case is the sarcoplasmic reticulum of the smooth muscle. This calcium then, by its release, so we can put this in here, we get calcium release. This calcium release causes smooth muscle contraction. So those are the two opposing effects in the autonomic nervous system. Epinephrine causes relaxation, and acetylcholine causes contraction. The epinephrine functions mostly when you're in sympathetic drive, so that you're now opening up those tubes, reducing the friction of the air against the walls, so it's easier to breathe in and breathe out. When you're sitting home quietly, you reduce the overall volume of the conducting zone by constricting the smooth muscle and reducing the diameter of those bronchioles. Let me finish this story by going back up here. And I'm going to remind you, I'm going to go back to the back board just here in a moment. I'm going to remind you that there's a continuous layer of endothelial cells all along the system, down into the tiny bronchioles. And I'm going to come back here and describe the epithelium. So this is the bronchiolar epithelium. It sits on a basal lamina. And outside that basal lamina, surrounding the tube, is going to be the smooth muscle. So the smooth muscle is the snake. Smooth muscle is going to snake around that bronchiole. Now, occasionally, within these, among these epithelial cells, there's going to be another cell type. And this cell type is called a mast cell. And if we look closely at the mast cell, it contains vesicles filled with histamine. In addition, we find that the apical surfaces of these mast cells frequently will contain on them antibodies. Usually IgE or IgA. These antibodies can cause problems because they can bind to an allergen. Among the most notorious of these are peanut lectins, which will bind to these and cause now a massive release of histamine. So we get the histamine release. In addition to the G protein coupled receptors, the beta 2s and the M3s, there's another G protein coupled receptor out here called a histamine receptor. And the histamine receptor will bind histamine. And this leads to smooth muscle contraction. And that's the basis of an asthma attack. That's where asthma comes from. You breathe in an allergen, and it binds to the IgA or IgE. The IgA or IgE releases histamine. The histamine causes smooth muscle contraction, and your bronchioles constrict, and you have a hard time breathing. Let's leave it with that. But what I want you to think about is from the story I told you, why do people carry EpiPens? EpiPens contain epinephrine. And why do nebulizers that you breathe in, why do they contain a beta-2 agonist that will activate that receptor? If you can follow through that logic, you'll see what's going on in this whole story. And I'll leave with that.